Hello lovelies, in this video we're going to carefully and calmly take you through everything you need to know for your foundation AQA GCSE Biology Paper 1, so you don't need to worry about the other videos that have higher tier content in. This is just everything you need for a foundation. If you want to revise something in particular, then you can use the timestamps in the description or the chapters to jump to the bit that you're looking for, but if you're doing this the day before the exam, just watch the whole thing through carefully and slowly. If you really want, you can put it on 2x speed, but we are going to take things calmly here. Now, after each section, there's a set of questions, set flashcards over on the website, so you can pause the video, go and do the questions, check that you understand everything, rewind if not, and then move on to the next section on the video. And then you can do the predicted papers and the walkthroughs of the predicted papers that we've got for this year as well. Because having an examiner explain to you how to lay out your answers and what sort of things an examiner is looking for in the questions that we think are going to come up this year is amazing revision.
So we need to be able to label a basic animal cell and to know what all the parts of the animal cell does as part of the function of the cell. So first up on the outer edge we have the cell membrane, then we have the cytoplasm. These little dots are representing ribosomes. You're very unlikely to need to be able to label ribosomes in a diagram in the exam. If they've got like lots of little dots all the way across the cytoplasm of the cell, that's just to kind of represent the cytoplasm. So it would be very specific and they look like special circles if they are talking about the ribosomes. Then we have the nucleus, which is the biggest circle-shaped structure inside the cell. And then we have the mitochondria. So these are normally little kind of oval shapes with wiggly lines inside. That's on a diagram how you would recognise that it's a mitochondrion. So the cell membrane's function is to control the entry and exit of substances into the cell. The cytoplasm is where chemical reactions happen inside the cell. The ribosome's function is to do protein synthesis, which means to make proteins. The nucleus's function is to control the cell activities. And the mitochondria's function is it's where aerobic respiration happens, and that provides energy for the cell. Now let's do the same with plant cells. So plant cells also have a nucleus. Plant cells also have ribosomes. They have a different oblong-shaped structure to mitochondria, which are the chloroplasts. Again, these will look different to mitochondria. They'll be oblong-shaped, but they often normally have little stacks of or disc shapes inside instead of wiggly lines. That's how you know it's a chloroplast. Then we have our mitochondria. There's normally a large space area, which is the vacuole or the permanent vacuole. Then the inner edge this time is the cell membrane and the outer edge is the cell wall. So I've filled in all of the functions of the parts that we've already looked at in the animal cell. For the new parts, the chloroplast's function is to absorb light energy for photosynthesis. This whole phrase is something you need to learn for the exam. The permanent vacuole contains cell sap, but its function is to be able to support the cell structure. And the cell wall is strong because it is made of cellulose, and that is also there to help the cell structure. If you're comparing animal and plant cells, there are only 3 differences, which we've already spoken about. So the 3 features that are found in plant cells that are not found in animal cells are the cell wall, the vacuole, and the chloroplasts. Otherwise, everything else is the same. Both animal and plant cells are classed as eukaryotic cells. The word eukaryotic is used to describe any cell that has a nucleus, and that inside the nucleus is where the DNA is kept. So because both plant and animal cells have a nucleus, they are both eukaryotic cells. Prokaryotic cells is a different type of cell. These cells do not have a nucleus. The name prokaryotic literally means before the nucleus. So they evolved before a nucleus became structure in cells, so they are without a nucleus. An example of a prokaryotic cell is a bacterial cell, and their DNA is just found in the cytoplasm floating around, not inside a nucleus. We need to be able to label structures in bacterial cells as an example of prokaryotic cells. So they also have a cell membrane, their inner layer around the edge. They also have cytoplasm. They have sometimes a different structure called a flagellum, but not all bacterial cells have this. They have little loops called plasmids. They have ribosomes. Their DNA or their chromosome is just floating in the middle in the nucleus, as we said. And they have a cell wall, but it's a bacterial cell wall that is not made of cellulose. So again, I've put on the functions for the parts that we're familiar with. For the flagellum, again, remember only some bacteria have this, not all, but it's used for moving around or swimming. So the plasmids are small extra loops of DNA that often contain useful genes, like antibiotic resistance. The bacterial cell wall is for structure, but also for protection. We have to be able to compare prokaryotic cells and eukaryotic cells. The main two differences are that there are no mitochondria or chloroplasts in prokaryotic cells, and there is no nucleus. One similarity with plant cells is that there is a cell wall in prokaryotic cells. However, the cell wall in prokaryotic cells is not made of cellulose, so there is still a difference there. These diagrams we've looked at are very basic cell diagrams, but most cells don't look like this. Cells change their size, shape, and internal or subcellular structures to carry out specific functions in organisms. These are known as specialized cells. There are some specific examples of plant and animal specialized cells that we need to know. Root hair cells are one example of plant specialized cells. You can see here I've labelled all the structures we were labelling on our basic plant cell, but there's some things that are different. No chloroplasts, this elongated shape, for example. All of this is to help the root hair cell carry out its function, which is to absorb water and mineral ions from the soil. Xylem and phloem are two other types of specialized cell in plants that we need to know. They're highly specialized because they're very different from normal basic cells, and they transport substances around the plant. That's their function. Phloem transports sugars and amino acids, and the xylem transports water. Some animal examples we need to know. So a sperm cell, again, I've labelled all the structures that we're familiar with from the basic cell, but you've also got the flagellum, which is allowing it to help it swim to the egg, which is one of its main functions. A nerve cell, again, very similar. We can label all of those similar structures from the basic cell that we can see, but the difference in the shape here, the really long axon, is to help transmit electrical impulses across long distances. And finally, muscle cells. These cells have lots of mitochondria. So although they look pretty basic, actually they contain a lot more of mitochondria than a normal basic cell because they need that to provide the energy to contract, which is their function. You should be able to recognize these specialized cells, but also be able to label all of the structures that we normally label in a basic cell on these cells that look slightly different. Cell differentiation. Cells become specialized through the process of differentiation. This is where different genes are turned on or off in each cell to change their shape or the number of subcellular structures that they have. And this is what allows them to become adapted to carry out a particular function. Undifferentiated cells, so cells that have not yet differentiated to become specialized, are known as stem cells. So you can see here I have a stem cell, and then that stem cell can go through the process of differentiation to produce red blood cells, or a neuron, or epithelial cells, or white blood cells, or muscle cells, or bone cells. It depends on what that process of differentiation looks like in the cell, determines what cell it's going to look like at the end. Differentiation is different between plant and animal cells. Plant cells mostly have the ability to differentiate throughout their whole life, which is what makes cloning plants possible, because you can cut off a piece of leaf or stem tissue, and those cells can differentiate into root cells, and you can create a new plant that way. Animal cells are not like that, and they do not have this ability. Most animal cells differentiate at an early stage, and once they're specialized, very few of them can change or become stem cells again. They just divide by mitosis to replace themselves. So an example here is why differentiation and mitosis is important in animals is because we start off as a fertilized embryo, and then that goes through mitosis to develop a ball of cells, but all of these cells at this point are stem cells. As the embryo starts to develop and grow into a fetus, cells go through differentiation to produce all the different cells and tissues that the body needs. Eyes, ears, brain cells, heart cells, muscle cells, bone cells, all of those will be produced through differentiation. And then once that's happened and you have all of the cells and tissues in the body that you need to have a full organism, then we just go through the process of mitosis in order to grow and get bigger. So to grow from a small fetus into the full-sized baby, and then from a baby into an adult. Microscopes. This is an optical or light microscope which you may have used in school. Initially they had mirrors which would reflect sunlight or lamplight, but now we have electric bulbs in the bottom, and the light travels up through a glass slide, through objective magnifying lenses, and into the eye of the person looking down the microscope. The reason that we have microscopes is because they magnify images. So what we're able to do is make a structure on a living material, such as cells, bigger and appear bigger than they actually are, so that our eyes are able to see them. We can calculate the magnification of a microscope by doing the image size, so the image that we see from the microscope, what we can measure in that, divided by the actual size of the object we're looking at in real life. So this is basically telling you how many times we have multiplied the size of the real object in order to be able to see it in the image. Optical and light microscopes are great, and they've been able to show us many things, but we're only able to see like the nucleus inside a cell and just about mitochondria at about 1,500 times. But really, that's the level of detail we can go to. Anything smaller than mitochondria, like ribosomes, we're just not able to see using a light microscope. Electron microscopes are one of the newer technology microscopes we have. They have much higher magnification and resolution ability than a light microscope. This means we can see more structures that are smaller than with a light microscope, and the reason is because the microscope has the ability to see separate objects clearly at high magnification, which is what is meant by resolution. They're really important in terms of our discoveries because we've been allowed, therefore, to see inside cells in way more detail, and even inside subcellular structures like mitochondria and chloroplasts. And even in the smallest of organisms like bacteria, we've been able to see detail inside. It's been an important development in understanding life on Earth. You need to be able to explain and describe how to carry out a microscope practical where you can view structures on a microscope slide. First of all, we need to prepare the slide. This example is going to be with onion cells. So you would add a drop of water to a microscope slide. You would take a very thin piece of tissue, in this case onion skin. It would need to be thin so the light can pass through it. We then need to stain it, in this case with iodine, so that we can actually see the structures inside the cells, like the nucleus and the cell wall. Then you would need to lower gently a cover slip on the top of your stained tissue to cover it before you put it on the microscope. So when you go to put it on the microscope, you would put it onto the stage. So your microscope slide goes onto the stage of the microscope, and it's held in place by stage clips. Then you would need to rotate the objective lenses to the lowest power first. This is normally about 4 times. Once you've focused, you can come back to the objective lenses to increase the power to magnify the image more. At low powers, you use the coarse focus wheel to focus the image so that it is no longer blurry. At this point, then you can go back to the objective lenses and rotate them in order to increase the magnifying power. And then at high magnifications, you're going to use the fine focus wheel to make your image focused at a higher magnification. Chromosomes. DNA is a long molecule that forms a twisted double helix. Sections of DNA are called genes. DNA is a really long molecule, so it has to coil up and coil up and coil up to form solid structures called chromosomes to fit inside the nucleus. Inside the nucleus, chromosomes are normally arranged in pairs. You can see them here in humans. There are 23 pairs of chromosomes in the nucleus of every cell in the body. Mitosis and the cell cycle. The cell cycle is the process that all cells go through to prepare for cell division and then to divide, and then it starts again as soon as they've divided. The majority of the cell cycle is spent in interphase, which is where the cell is preparing for division. So it replicates its DNA, so it doubles it. It grows in size, you can say the cell elongates, and it increases the number of subcellular structures. So as well as doubling the DNA, the numbers of things like ribosomes and mitochondria will also increase. After interphase, the cell is ready to divide, and that process is known as mitosis, which is where actual one cell divides into two identical daughter cells. Firstly, the chromosomes are pulled apart to opposite poles or ends of the cell. The cell membrane and cytoplasm then divides. This forms two genetically identical daughter cells that have the same chromosome number. This process of cell division allows organisms to grow, so from small organisms to large organisms as they grow older, and also to replace cells in damaged tissues or organs. Stem cells. Stem cells are undifferentiated cells which can produce different cell types through the process of differentiation. We've looked at these already before. There are three different types of stem cells or sources of stem cells that we have to know. The first is embryonic stem cells. These can differentiate into nearly any type of animal cell. Adult stem cells, which can be found in bone marrow, can differentiate into some type of animal cells, for example blood cells, but not all types of animal cells. Plant stem cells are found in the tips of roots and shoots. These are known as meristems. They can differentiate into any type of plant cell. We mentioned before that plant stem cells can have this ability to differentiate throughout the whole of their life, and this is how we can use it to clone plants. This means we can make lots of plants really quickly, and that's useful in some cases because we can save rare species from extinction. Therapeutic cloning can be used to produce embryonic stem cells that are genetically identical to the patient. These stem cells can now be used to grow new cells that can replace the damaged ones that the patient needs. For example, blood cells after maybe a cancer treatment. To treat paralysis, we can grow nerve cells or nerve tissue when that's been damaged in the spine, for example, or pancreatic cells to treat type 1 diabetes. There are some ethical issues around using embryonic stem cells. There is an objection to the fact that the embryo cannot consent to be used, unlike if adult stem cells are taken from a patient that is able to consent. And the concept of an embryo being a potential life means that unused embryos being destroyed is not accepted by some people. There is also a risk of viral infection transfer, especially if the stem cells are coming from another person, or potentially a cancer, because stem cells can divide rapidly, so inputting dividing cells into the body could lead to a tumour. Diffusion is the net movement of particles from an area of high concentration to an area of low concentration. So net just means overall. So the particles will move randomly in random directions, but overall, over time, more particles will move from an area of high concentration to an area of low concentration. This is a passive process because it does not require energy. In terms of what's diffusing into and out of cells, mostly oxygen and glucose will be diffusing into cells, as these are needed for respiration, so they get used up and will be in low concentration inside cells. Carbon dioxide and urea diffuse out of cells. These are waste products from metabolic processes, so they will build up inside cells and be in higher concentration, so they will diffuse out. There are 4 factors that affect the rate of diffusion. One is the concentration gradient. If you increase the concentration gradient or difference in concentration, then you increase the speed of diffusion. If you reduce the distance that particles have to travel, that increases the speed of diffusion. Mostly this is done by reducing the membranes needed to cross or pass through in order to be able to get from one place to another by diffusion. Increasing the temperature increases the rate of the movement of particles, so that increases the speed of diffusion. And increasing the surface area of the membrane that particles have to cross also increases the speed of diffusion. There's more space for them to get across the membrane. All exchange surfaces will have adaptations to maximize the rate of diffusion. So exchange surfaces are places in organisms where majority of diffusion happens, and these are why they have this adaptation. They will all have thin walls, a large surface area, and good blood or air supply. Thin walls, such as having 1 cell thick walls in the small intestine in the villi, helps to reduce the diffusion distance and therefore speed up diffusion. A large surface area, obviously we know, increasing the surface area increases the rate of diffusion. And a good blood or air supply is ordered to maintain a steep concentration gradient. So constantly substances that are diffusing through are being moved along to create a low concentration gradient behind them. That means that diffusion keeps occurring in the right direction that we want it to happen. There are 3 specific exchange surface examples that we need to be able to explain and describe how they have these adaptations and how it makes them good at doing exchange. So for example, the alveoli in the lungs in humans, this is where gases diffuse between the lungs and the blood capillaries. The villi in the small intestine, this is where small molecules, so amino acids, glucose, fatty acids and glycerol, which are the products of digestion, diffuse from the small intestine into the blood in capillaries. And finally, leaves in plants. This is where gases will be diffusing in and out of stomata and into the spongy mesophyll tissue layers, where they will be exchanged with cells that are carrying out respiration and photosynthesis. This is all linked to this idea of surface area to volume ratio, which we need to be able to not only calculate, but explain. So surface area to volume ratio is where we divide the surface area of an organism by its volume. And we've gone with cubes here to give you an example, but the idea is that the larger the organism becomes, the smaller their surface area to volume ratio becomes. If you look at my cubes, and if we think about diffusion, the distance between the centre of each cube and then to the outside or to the external environment is much greater in the larger organism with the larger length of size of the cube. So compared to its volume, the distance between its volume and all of its outer edges is longer. This means diffusion from the very inside of that organism to the very outside of that organism would be very slow because the distance is very long. So we can't just rely on substances diffusing from the outside into all of the cells in the body if we are a larger organism. Single-celled organisms, like this example I've got here, do not need an exchange surface like the ones we've just looked at, because they have a very large surface area to volume ratio, because it's a short distance between all of their internal sections to their external surface membrane. So it's really quick for substances to diffuse from outside of the cell to the inside of the cell, and therefore they don't need to have any adaptations to speed up that rate of diffusion. However, the larger an organism gets, the smaller its surface area to volume ratio becomes. So it needs these specially adapted exchange surfaces like alveoli, like villi, and then a transport system like our circulatory system in order to increase the rate of diffusion of substances into and out of every cell in the body, so that it can happen fast enough for processes like respiration and photosynthesis to occur. Osmosis is the net movement of water molecules from a dilute solution to a more concentrated solution through a semi-permeable membrane. It's really important when you're writing this definition in the exam that you say water molecules, because it's only water that moves by osmosis, and that you get the dilute and concentrated the right way round, and you don't forget to add the part about a semi-permeable membrane. This could be a 3-mark definition, so it's important to have all of that learned as part of your definition for osmosis. When we say a semi-permeable membrane, what we mean is a membrane that lets small molecules through but not large ones, for example, a cell membrane. You can get artificial semi-permeable membranes as well, and they can use those as an example in the exam, so it's not always something that happens just through cells. Just like diffusion, it's a passive process because it does not require energy. Water will move into or out of cells by osmosis depending on the difference in concentration of the cytoplasm of the cell, so inside the cell, compared to any solutions outside of the cell. Just like diffusion, it's affected by the same factors: temperature, surface area, and concentration gradient. If you increase the temperature, or if you increase the surface area, or you increase the concentration gradient or difference, then faster osmosis will occur. There are 3 conditions, and we have to know what would happen to animal and plant cells in each of these conditions. Cells that are in a dilute solution, such as pure water, water will move into cells by osmosis because there is more water outside of the cells than in the cytoplasm of the cells. Animal cells can swell and they can burst. Plant cells will swell up, but they don't burst due to their cell wall stopping that from happening. When cells are in a solution that has the same concentration as their cytoplasm, there will be no net movement of water into or out of cells. This means that no osmosis will occur. Cells remain in the same state in terms of how much water is inside their cells. This is why it's important for things like blood plasma, which is the solution surrounding red blood cells, to have the same concentration as red blood cell cytoplasm. When cells are placed in a more concentrated solution, such as salt or sugar solution, water will move out of the cells by osmosis. Animal cells can shrink and shrivel up. In plant cells, they don't shrink and shrivel up as much because their cell wall is very strong and stays pretty much in its shape. But the vacuole will shrink and the cytoplasm can pull away from the cell wall in extreme circumstances. Now you'll notice in each of these I've used the same phrase about where water is moving to and from, and I've used the phrase by osmosis. That is really important. If you are answering an osmosis question in the exam, you have to use the word osmosis when you're describing not only where the water moves from and to, but how it moves, as in by osmosis. We need to be able to explain how we can do practical methods in order to observe osmosis happening. So this is an example, and most of the examples you'll see are where we use different concentrations of either salt or sugar solution, and we put a piece of plant tissue into that solution. This can be any plant tissue: aubergine, potato, red pepper, carrot. It doesn't matter what the tissue is, as long as it's got plant cells, because these cells will not shrivel, or burst. We want them to just change in terms of their mass. And then we have to make sure that we keep all of our control variables correct to make sure that we're just observing what's happening to those cells as a result of being in the different concentrations of the solution. So in nearly every example of this practical, your independent variable is the concentration of solution, because that is what is being changed. Your dependent variable is the mass of tissue, or the change in mass of tissue from before and after the experiment. This makes sense because if cells gain water by osmosis, they'll increase in mass, and if they lose water by osmosis, they will decrease in mass. There are many control variables in this experiment. For example, the volume of solution. The time that the plant tissue spends in the solution should be the same for all the different concentrations. The length and volume of the tissue pieces, so how you cut them, they should all be the same size, but rather than saying size, use the words length and volume, because we're trying to make sure that they have the same surface area to volume ratio, because that can affect the rate of osmosis. The same type of tissue should be used, so from the same potato or from the same type of potato, for an example, so that there isn't differences between the cytoplasm of the cells. There should be no skin on any pieces of tissue, because skin is made up of a different type of cells, and this can be waterproof, so this can affect osmosis. The temperature of the solution should also be the same. You can either do this experiment in a water bath to keep it the same temperature, or at room temperature is fine. Remember, control variables should be anything that could affect the results of the experiment, and in this case, it's anything that could affect the rate of osmosis in each of these different solutions. The other key thing to remember is that when we are weighing and measuring the mass of the tissue, after it has been in the solution, we need to blot our pieces of tissues dry to remove any solution so that doesn't affect the mass. I've got an example of a graph produced from data from this experiment, and always need to remember that when we are looking at graphs like this, often they will ask you to show the concentration of the tissue. How could you use this graph to work out what the concentration of the cytoplasm of the cells in these pieces of plant tissues is? So if you plot percentage change in mass against the concentration gradient, and then you look for where the line of best fit crosses the x-axis, which will be 0. So at that point, there is 0 change in mass. So we know that there's no water moving into or out of the cells. Therefore, in this case, the concentration that is closest to the cytoplasm of the cells in this example is 0.6 molar. So this is how you would use these graphs. It's always looking for where the line of best fit crosses the x-axis at 0, where that means there is no change in mass. Because that must mean that the concentration of the solution and the concentration of the cytoplasm of the cells is the same, so there is no osmosis. Active transport is the movement of particles from low to high concentration against their concentration gradient using energy. This is not a passive process, unlike osmosis and diffusion, because it requires energy from respiration. Unlike osmosis and diffusion, the rate of active transport is not going to be affected by the same factors. It's mostly affected by the rate of respiration. As the rate of respiration increases, the rate of active transport increases. This means that it can be affected by the number of mitochondria in a cell or the availability of oxygen. If this isn't there or present, then less active transport is going to be able to take place because there'll be less respiration happening. It can also be affected by the number of carrier proteins. So the reason active transport requires energy is because we're using proteins in the cell membranes to actively change shape and move molecules from one side of the membrane to the other. We need to know 2 examples of where active transport occurs in cells. Plants, it's the root hair cells. These transport mineral ions against their concentration gradient from the soil into the root hair cell. The animal example can be found in the cells that are around the villi, which line the small intestine. These transport glucose molecules against their concentration gradient from the small intestine into the blood after equilibrium is reached. Both of these cells also carry out diffusion, but having this ability to do active transport maximizes the absorption of these essential nutrients. So plants cannot survive without mineral ions, and in animals we need all the glucose we can get from our food in order to carry out respiration. So once diffusion has happened and these molecules have tried to move into cells down their concentration gradient, if there is still glucose left in the small intestine, and if there's still mineral ions in the soil, but there's a high concentration already inside the blood or inside these cells, they will use active transport to make sure they get every single last mineral ion or molecule of glucose to make sure that we absorb the maximum that we can. The adaptation of these cells is that they will have lots of mitochondria to help provide the energy needed for lots of active transport. Any cell that you get told or is given as an example of a cell that carries out a lot of active transport will require a lot of mitochondria to provide enough energy through respiration to do that process. We need to know how living organisms are organised, so from the smallest part all the way up to the whole organism. Cells are the smallest living unit and the most basic building blocks of life. A group of similar cells working together to carry out a function is known as a tissue. A group of tissues working together to carry out a function is known as an organ. Then groups of organs come together to form organ systems. Multiple organ systems work together to form the whole organism. So an example of a tissue is a palisade tissue layer in leaf, which carries out photosynthesis. An example of an organ is the heart, which pumps blood around the body. An example of an organised system of organs is the nervous system. And then a whole organism would be something like a whole plant, a whole insect, or obviously a human. And so we need to be able to understand that these go in size order as well. So cells are the smallest, then tissues, then organs, then organ systems, then the whole organism. Here's the diagram of the labelled digestive system. You should hopefully know this from Key Stage 3 and be able to label one of these exactly like this yourself. The salivary glands in the mouth secrete saliva that contains the enzyme amylase. This digests carbohydrates. So an example is starch, which would be the substrate. The enzyme is amylase, and they react together and it breaks down starch into glucose, which is smaller sugars. The stomach contains hydrochloric acid to kill bacteria and secrete protease enzymes to digest proteins. So this is where we have proteins. The protease enzyme breaks these proteins down into the smaller molecules, which is amino acids. The small intestine is where the absorption of all the small molecules into the blood occurs. It contains all three types of enzymes that are secreted by the pancreas, including lipase to digest lipids. So lipids are broken down by lipase enzymes into glycerol and fatty acids. So on this page, we need to be able to remember all of the enzymes, what they break down, what they break their substrate down into, so the product or the small molecule. We need to know where they are secreted from, which I've written down here, and where they work. So in the salivary gland is where amylase is made and it works in the mouth. Protease is made in the stomach and it works in the stomach. And then the pancreas makes all three enzymes, a form of amylase, proteases, and lipase. So it's made in the pancreas, but they work in the small intestine to complete digestion, and that's then where all of those small molecules are absorbed. When we're talking about enzymes and how they work, we need to understand that they work through this idea of the lock and key mechanism, where the substrate acts like a key and the active site of the enzyme acts like a lock. It has a specific shape so that only one substrate can bind to the enzyme. Digestive enzymes are really important because they break down large insoluble molecules to small soluble molecules which can be absorbed through the cell membranes and into the blood to be transported and built into new molecules or for respiration in all the cells in the body. Amino acids, the small molecules that are absorbed, can be used to build proteins again for growth. Glucose can be used to build other carbohydrates like glycogen as an energy store, or they can be used directly in respiration to release energy for cells. The fatty acids and glycerol can be used again to make lipids, which can act as an energy store, or they can act as insulation. The rate of enzyme action, or the rate of enzyme-controlled reactions, can be affected by a few things. One of them is temperature. If you increase the temperature, you'll increase the rate of reaction, unless it gets too high and then the enzyme will become denatured. pH is also a factor that affects enzymes. Outside of the optimum pH for an enzyme, it can denature and therefore slow down the rate. And surface area of a substrate or the concentration of a substrate, if you increase this, it will increase the rate of reaction because there's more substrate and more surface area for the enzyme to bind to. When we say denatured, what we mean is the active site changes shape and therefore it can no longer bind to the substrate. The last part of digestion we need to know is about bile. Bile is produced by the liver and it neutralizes stomach acid in the small intestine. It also helps to digest fats through the process of emulsification. So it is made in the liver, it is stored in an organ called the gallbladder, which then releases it into the small intestine, which is where it works to help digest fats. Bile is not an enzyme. Increases the speed of digestion of lipids, but it does not actually do the breaking down. It is not an enzyme. It just binds to large lipid droplets, breaks them into smaller lipid droplets, which increases the surface area so that the lipase enzyme can bind to a greater surface area and therefore break down the lipids into glycerol and fatty acids faster. For all those molecules we talked about during digestion, we need to be able to show how we can identify these different molecules in food using various chemical tests. For starch, you would add iodine, and if starch is present, there'll be a colour change from yellow-brown to blue-black. You must say blue-black, or you can say black in the exam, but you cannot just say blue, because this is the colour of other test reagents. For proteins, you would add biuret reagent. The colour change, if protein is present, is from blue to purple. For glucose, you would add Benedict's reagent. We must spell this with a capital letter, same as biuret, because they are named after people. And you must heat it up. It will not work at room temperature. You should say heat, boil, or specify it's over 80 degrees. If glucose is present, there'll be a colour change from light blue to brick red. For lipids, you would add ethanol, then add water and shake to mix it together and a cloudy white emulsion will form. A negative result for any of these tests would be no colour change. So staying yellow-brown for starch, staying light blue for protein and glucose, and then no emulsion present for lipids. There are many ways that you can measure the rate of an enzyme reaction, and you could be given lots of different examples in the exam. The independent variable could be pH or temperature or substrate concentration, because changing all of these can affect the rate of an enzyme reaction. Depending on what the reaction is and what you're measuring, it's going to change the dependent variable. So you could measure a colour change using an indicator, so the time it takes for a colour to change to indicate that a substrate has been used up. You could measure the mass or volume of the product of an enzyme reaction. You could measure a change in pH. So for example, when lipids break down, they produce fatty acids. So the pH will decrease as more fatty acids are produced over time. So you can measure this with a pH meter. There are also control variables for each experiment, but similar ones will be things like volumes of solutions, the concentrations of solutions, and then pH or temperature, whichever one you are not changing as your independent variable. The example you may have looked at is starch and amylase. In this example, our independent variable is temperature, and we can use a water bath to vary the temperature of the different reactions. Our control variables would be using the same concentration and volume of both the substrate and the enzyme. And then we can measure the time that it takes for iodine to stop changing blue-black to show that all the starch is digested. So taking small samples of the mixture solution every 30 seconds, testing it with iodine until it no longer turns blue-black to show that all of that starch has been digested by the amylase enzyme. This is a graph of something predictable, like a similar result that you might see, where we've got the temperature on the x-axis and the rate of reaction on the y-axis. We normally see this mountain shape, where we can describe that as the temperature increases, the rate of reaction increases due to the increased kinetic energy of the particles. There's a peak, which is the optimum temperature, and then after the peak, the rate of reaction decreases as temperature increases because the enzyme is denatured. You'll notice that most human body enzymes, like the ones we've been looking at for digestion, will have an optimum temperature around the same as human body temperature, which is about 37 degrees C. We need to know the 3 different types of blood vessels and how their structure relates to their function. Arteries transport blood away from the heart at high pressure. This is how I remember the A for arteries and A for away from the heart. They have a narrow lumen to maintain high blood pressure. They have a thick elastic layer so they can stretch and recoil to maintain high pressure. So the maintaining high pressure here is part of their function, and this is how it relates to that bit of structure. So if you're describing and explaining the structure and function of arteries, you need to talk about the narrow lumen is a structure. What does it do? How does it help it carry out its function? It helps to maintain high pressure. Lastly, it has a thick muscle layer to withstand high pressure. You need to use these words, otherwise you won't get marks. Veins transport blood to the heart at low pressure. They have a wider lumen to transport a greater volume of blood. They have a thinner muscle and elastic wall layer because their blood is being transported at lower pressure. So we do not need the same features we see in arteries. Veins have valves. I remember this with the V for vein and the V for valves. This is to prevent the backflow of blood due to gravity. Capillaries transport blood close to cells so that substances can be exchanged. They have a very narrow lumen to slow blood flow down to one blood cell at a time. This increases the time for diffusion to occur. They have thin walls that are only one cell thick to decrease the diffusion distance. And ultimately they join small arteries and veins together So once exchange has happened, the blood flows into veins and then can return back to the heart. You need to be able to use all of this information to be able to compare the structure of the different blood vessels as well as their functions. Blood is a tissue which transports substances around the body and plays a role in the immune system. The majority of blood is made up of blood plasma, which is the liquid that blood cells are suspended in. It's mostly water, but it contains all the dissolved substances that are being transported around in the blood. Things like glucose and other things like antibodies, for example, and other dissolved substances from the digestive system, like the amino acids and things that need to travel to and from your cells. The rest of the blood is made up of 3 different types of blood cells that are suspended in this liquid. They're red blood cells, white blood cells, and platelets. Platelets are involved in clotting open wounds to try and prevent infection or to prevent excess bleeding. Red blood cells are specialised cells because they have no nucleus. They contain lots of the hemoglobin protein, which is the red coloured protein that binds oxygen. They have a biconcave shape, which gives them a large surface area to increase diffusion of gases into and out of the cells. And their job is to transport oxygen to and from cells. There are 2 types of white blood cells: phagocytes, these engulf pathogens, and lymphocytes. These produce antibodies or antitoxins. Both of these defend the body from pathogens and prevent you from getting sick, so they play a part in the immune system. We need to be able to label and describe the structure of the heart. First of all, with any heart diagram, always write on the right and left, the right way round, so it is the opposite to how your hands are on the page. Then we won't get mixed up with our rights and our lefts. The 2 top chambers are the atrium, and the 2 bottom chambers are ventricles. Think about it as A comes before V in the alphabet, so A is at the top and V is at the bottom. The blood vessel that brings deoxygenated blood from the body to the heart is the vena cava. It is a vein. Once blood has entered the right atrium, it's going to flow down into the right ventricle, and the contraction of the right ventricle will push the blood up and out of the heart through the pulmonary artery. It is an artery because it's taking blood away from the heart, and it is taking blood to the lungs to be oxygenated. When blood returns to the heart from the lungs, it returns in the pulmonary vein. So because it has been to the lungs, it is now oxygenated blood. This is the only vein in the body that carries oxygenated blood. The blood then moves through the left atrium, which contracts and pushes it into the left ventricle, which will contract and push it up and out of the aorta. This is the biggest artery in the body, and it takes blood at the highest pressure because it has to send it to the whole of the rest of the body. The left ventricle has a thicker muscle wall. This is so that it can contract with greater force, so that blood is forced out through the aorta at higher pressure, because it has to go a further distance to get around the whole body than when it leaves the pulmonary artery and just goes to the lungs. The human circulatory system is known as a double system or a double pump system because the blood flows through the heart twice, once to the lungs and once to the rest of the body. So the right side sends blood from the body to the lungs, and the left side sends blood from the lungs to the rest of the body. The heart rate is controlled by a group of cells known as the pacemaker cells that are found in the right atrium. If these cells stop working, they can cause arrhythmia, which is an irregular heartbeat, and an artificial pacemaker can then be fitted to control and regulate the heartbeat. The right-hand side of the heart pumps blood to the lungs to be oxygenated and to remove carbon dioxide. You can see a diagram of the lungs here. We should know the trachea, or the windpipe, branches out into 2 bronchi, and then the alveoli at the end of the tiny branches, which are known as bronchioles, and we have 2 lungs, one on either side. Here we've got a diagram of one alveolus from a bunch of alveoli and the capillary that's next door. You can see both of the walls are only 1 cell thick, as we mentioned, as this is an adaptation. We have deoxygenated blood coming from the heart on the left-hand side. When it reaches the alveolus, then diffusion of carbon dioxide from the blood into the alveoli will happen, and then oxygen will diffuse from the alveoli into the blood. This is where it will bind to the red blood cells, which will carry it and transport it back to the heart. Once the oxygenated blood will arrive back at the heart, then the left-hand side of the heart will pump this to the rest of the body. Coronary heart disease. The coronary arteries are blood vessels which transport blood to the heart muscle itself. If these coronary arteries get blocked with a fatty buildup called plaque, then blood flow is reduced, so less oxygen and glucose can reach the heart muscle. Without enough oxygen and glucose, the heart muscle cannot respire, so it stops contracting. This can cause a heart attack. Some risk factors for coronary heart disease, that means they increase the risk or the chance of you getting coronary heart disease, are poor diet and lack of exercise. This is because they can lead to other conditions such as high blood pressure, high cholesterol, and obesity, which are all medical risk factors for coronary heart disease. We need to be able to describe some treatments for coronary heart disease. Statins are drugs that can be taken to lower cholesterol. These are a preventative measure, so they cannot reduce any buildup that is already there, but they can reduce future buildup of plaque or fat in the coronary arteries in the future. Stents are a metal mesh-like cage which is used to widen arteries that have been narrowed by this fatty buildup. This can help to increase blood flow. With serious coronary heart disease, it can cause heart failure. If heart failure has been caused, then the options for treatment are a heart transplant or a heart bypass, or an artificial heart, which can be used to keep someone alive until a transplant becomes available. You need to be able to discuss the advantages and disadvantages of these different types of treatments for coronary heart disease. Health is a state of physical and mental well-being. Causes of ill health include disease, which can either be communicable disease or non-communicable disease. Communicable diseases are diseases that can be passed from one person to another, and they are caused by a pathogen. Non-communicable diseases cannot be passed from one person to another. Diet, because this can lead to certain non-communicable diseases. And stress, which can be caused by social or lifestyle factors. Different types of diseases might interact with each other, and you need to know how. Viruses can infect cells and cause cancer. For example, HPV can cause cervical cancer. Immune reactions to infections with a pathogen can trigger allergic reactions like skin rashes and asthma. So asthma is a form of allergy. Severe physical ill health caused by disease can lead to depression and other mental illnesses. Non-communicable diseases cannot spread between organisms. They're not caused by pathogens. They are usually long-term and get worse slowly. Examples include asthma, diabetes, and heart disease. Risk factors can increase the chance of getting these diseases. Non-communicable diseases can be caused by genes or genetic factors. These are out of a person's control. They could be caused by a mutation, or they could be inherited from their parents. The following are all risk factors related to lifestyle and often choices. Alcohol has been linked to effect in unborn babies' brain development. Excessive alcohol consumption has also been linked to liver disease and brain damage. A high-fat diet can lead to obesity. Obesity is a risk factor for type 2 diabetes, and a high-fat diet can also lead to high cholesterol, which is a risk factor for heart disease. Smoking whilst pregnant can reduce the birth weight of unborn babies. Smoking has been directly linked to causing lung cancer and lung diseases, and it also increases the chance of heart disease. Ionizing radiation, this is a link to your physics specification. So a carcinogen that damages DNA in cells, so it's an example of that. Exposure to ionizing radiation has been linked to cancer. For example, UV ray exposure has been linked to an increase in skin cancer. So you've got this idea that ionizing radiation will damage DNA, and then that can lead to it causing cancer, which we're going to look at next. But most diseases will be caused by a mixture of different factors coming together. It's not always just one of these. Cancer is a non-communicable disease where uncontrollable cell division causes tumours to form. There is some damage to the cell, normally to the DNA. This alters the way the cell division process occurs. This leads to uncontrolled or unlimited cell division, which then forms a tumour. Some tumours can be classed as benign. These are slow-growing and non-cancerous because they are contained in a membrane and do not spread to other parts of the body or other tissues. Malignant tumours, though, are cancerous. They are fast-growing and they can break off and spread to form secondary tumours in other parts of the body or other tissues while travelling through the bloodstream. You need to be able to define cancer as uncontrollable cell division and know about the two different types of tumours. Risk factors for cancer. Smoking introduces carcinogenic chemicals into the body through the tar that's in the cigarettes, and it can cause lung cancer, but others as well. Exposure to UV radiation, which we said is a form of ionizing radiation, has been linked to skin cancer. Obesity is a risk factor because we know that fat cells can signal other cells to divide. Viral infection, we've talked about the example with HPV that can cause cervical cancer. Some cancers can be caused by genetic factors, so either mutations in DNA or inherited genes that can increase your chance of getting a certain cancer. For example, the BRCA gene, which is known to increase your chance of getting breast cancer. A leaf is a plant organ that is made up of different tissue layers. One of the tissues is the xylem. This transports water and mineral ions. The palisade mesophyll is a tissue layer that contains cells that have lots of chloroplasts in order to absorb light for photosynthesis. It's where the majority of photosynthesis happens in a leaf. The upper epidermis is a transparent layer of tissue that lets light through to the palisade layer for photosynthesis. The spongy mesophyll tissue is an open tissue with lots of air spaces between the cells, creating a large surface area for gases to diffuse, and it's where the majority of gas exchange takes place. The lower epidermis contains the stomatal pores that allow gases and water vapour to enter and exit the leaf. The phloem is the tissue that transports sugars. The guard cells are specialised cells in the layer of the lower epidermis. They control the opening and closing of stomata. They must balance the need to open the pores for gas exchange with trying not to make sure that they're open too often or for too long to prevent too much water also being lost through transpiration through the pores. The roots, stem and leaves form an organ system in plants which transport substances. The main method of transport here is transpiration, which is the flow of water from roots to the leaves and is known as the transpiration stream. The process of transpiration is where water vapour evaporates through the stomata. The xylem is a tissue made of specialised cells that are adapted for the transport of water and mineral ions. Water flows up one way from the roots to the leaves through the transpiration stream, through the xylem, which is found in the roots, the stem, and the leaves. They have thickened walls of lignin to help them cope with this transport function, and their cells have no end walls between them, which allows this continuous column of water. Phloem is a tissue that contains specialised cells that are adapted for transporting the products of photosynthesis, mostly sugars, but also amino acids, around the plant, from the leaves to everywhere else in the plant that might need them. This process is known as translocation. The main adaptation of phloem cells is that they are elongated and they have little holes or perforations in their end walls to allow substances to pass through freely. Transpiration is the evaporation of water vapour through the stomata of leaves. Transpiration, or the rate of transpiration, can be measured using a potometer. So you can see the diagram set up below. This is just an example. Different potometers can look different, but we need to understand that we have a plant shoot, which will have leaves and stomata that the water can evaporate from, and that is connected all the way with an unbroken column of water from the bottom of the stem through a capillary tube, which has an air bubble in it. That's the only air bubble though. And we can move the air bubble to the start of the scale with the tap. And the air bubble is next to the scale, so we can measure the distance that the air bubble moves as water is sucked up through the plant shoot. And we are relating the fact that if it's sucking up more water from the plant shoot at the bottom, that is because more water is being lost at the leaves. You need to be able to explain how different factors can affect the rate of transpiration. Air movement or wind, if this is increased, it will increase the rate of evaporation of water, so therefore increase the transpiration rate. Light intensity, if this is increased, more stomata will open, so therefore this increases the transpiration rate. Temperature, if this increases, then the rate of evaporation is increased. Humidity, this is the only one that does the opposite, and transpiration will decrease as humidity increases because it decreases the rate of evaporation. Communicable diseases can spread from one organism to another because they are caused by pathogens that can be transferred. Pathogens are microorganisms that cause disease. One example of a pathogen is a virus. These are small, acellular, which means they are not made of cells. They live inside cells and replicate, causing damage to the cell when they burst out. They can reproduce rapidly inside humans. Another example of a pathogen is bacteria. These are prokaryotic cells that can reproduce rapidly inside humans. Bacterial cells release toxins which cause damage to cells inside the body, which is what gives you the symptoms and makes you feel sick. Another example of a pathogen is fungi. These can grow on the surface of leaves or skin, and they feed off living tissue. And lastly, protists. These are single-celled and often parasitic organisms that live inside and replicate inside host organisms, which cause damage. Communicable diseases can be spread through the air, through direct contact, or through vectors. An example of transmission by air is known as droplet infection. This is where we inhale pathogens in particles from coughs and sneezes. There are two forms of direct contact, either by consuming contaminated food or water that contains the pathogen, or by touching or having contact with infected tissue, for example, blood. An example of contaminated food is Salmonella, which is a bacteria that causes food poisoning. And an example of direct contact through infected tissue is HIV, which can be transmitted through bodily fluids, including blood. An example of vector transmission is malaria. So this is caused by a protist pathogen, which is transmitted by a mosquito vector when it bites humans. There are three examples of viral diseases that we have to know. The first one is measles. The symptoms of measles are kind of a red rash, so spots on the skin, and a high temperature or fever. It's spread through droplet infection because it also causes a cough, and it can cause blindness and even death, especially in children. The only way to prevent the spread of viruses like measles is through vaccinating young children. The next example is HIV. This is a virus that infects and kills white blood cells. It weakens the immune system and eventually leads to AIDS, which is where a person has a weakened immune system and will get secondary infections such as pneumonia or colds or bacterial infections, which can then kill them because their immune system is too weak to be able to fight them off. This is transmitted through sexual contact or bodily fluid contact. So one way of preventing the spread is by using condoms as a barrier method of contraception. The tobacco mosaic virus is an example of a plant viral disease. It infects plants and it destroys their chloroplasts in the leaves. This creates a mosaic pattern of discoloration on the leaves and reduces the plant growth due to lack of photosynthesis. You can prevent the spread of the tobacco mosaic virus by burning the whole plant and disinfecting any tools or hands that have touched the plant, and it can be spread by insect vectors such as aphids, so killing those can also help prevent the spread. There are two bacterial disease examples we need to know. The first is gonorrhea, which is a sexually transmitted infection, so we can prevent the spread with condoms, similar to HIV. The symptoms caused are discharge from the genitals and pain when urinating, and it can also cause a fever. Salmonella is the second example. This causes food poisoning that we have already mentioned, as it's spread from eating contaminated food, for example, raw chicken. The symptoms are sickness, fever, and diarrhea. We can prevent the spread by washing our hands before and after touching raw meat, and also cooking food to high enough temperatures to kill the bacteria. In the UK, chickens are also vaccinated against salmonella to try and reduce the spread and make sure it's not present in eggs as well. In both of these examples, the symptoms are caused by toxins released from the bacteria. Treatment for any bacterial infection involves antibiotics. These are drugs that kill bacteria in the body or stop them from replicating. Not all antibiotics will be able to kill all bacteria. We now have an example that there are many resistant strains of gonorrhea in the UK specifically, which cannot be treated with most antibiotics. There is one fungal disease example that we need to know that infects plants, and that is rose black spot. The symptoms are suggestive of the name, so black or purple spots on the leaves, and the leaves can also turn yellow and fall off. Plants often suffer from lack of growth because of lack of photosynthesis, due to these black spots and yellow areas, meaning there's less chlorophyll. It is spread through fungal spores that travel by wind or water, mostly in rain droplets. They can also be spread on gardeners' tools and hands as they work between plants. One way of preventing the spread is to move an infected plant away from the others until it is treated and safe to move back close to them. We can spray plants infected with rose black spot with fungicide in order to kill the pathogen. We can also remove infected leaves and burn them to kill the pathogen and its spores. These two will both help to prevent the spread of rose black spot to other plants. There is only one example of Protist disease that we have to know, and that is malaria. Malaria is caused by a Protist parasite that can live in both mosquitoes and humans, so some of its life cycle is spent in both organisms, and it can cause cycles of fever and it can be fatal. Ways to prevent the spread of malaria include preventing yourself from being bitten. This could mean using mosquito or insect repellent or mosquito nets. Or we can prevent mosquitoes from breeding to reduce their numbers by covering standing water, which is where they lay their eggs and their larvae develop. Human defense systems. Humans have some non-specific defenses that try to prevent pathogens from entering the body. The nose contains hairs and mucus to try and trap pathogens. The trachea and bronchi in the airways contain cilia on the cells to move mucus, which contains trapped pathogens, to the nose and mouth to be removed from the body, or to be swallowed. The stomach contains hydrochloric acid to kill bacteria in food or swallowed mucus. And the skin acts as a physical barrier to prevent the entry of pathogens everywhere else. Specific defences work inside the body in case pathogens do enter, and they're normally carried out by white blood cells. Phagocytes detect and engulf pathogens and digest them with enzymes. Lymphocytes are triggered to divide by mitosis when they detect a foreign antigen. They will form plasma cells, which release antibodies that are specific to the antigen on that pathogen, and they can also produce antitoxins that can bind and neutralize toxins released from bacteria. Vaccines contain a dead or weakened form of a pathogen, so they will not infect the person, but the antigens on the pathogen will bind and trigger white blood cells to produce antibodies that can bind to the specific antigen on the pathogen. So you can see I have my pathogen, my antigen, which is kind of the spiky protein on the outside, and my purple antibodies, which can bind to that antigen. The white blood cells will form memory B cells that will remember the antigen on the pathogen and will produce more antibodies faster next time they are reinfected in order to prevent illness. This makes people immune to that pathogen. So the first time you are infected with pathogen or an antigen from a vaccine, we go through the primary immune response to make antibodies for the first time. So not many antibodies are made, and it happens slowly. The second time, you get infected with the same antigen. So if you inhale it naturally, for example, through droplet infection, we have what's called the secondary immune response. So because you have memory cells, they produce the antibodies faster and there's more of them. And so we get that bigger peak in the graph that we can see here for the secondary immune response. This prevents you from getting sick and gets rid of the pathogen from the body before it has a chance to make you ill and before you can pass it on to other people. Vaccinating large numbers of a population can prevent the spread of a disease because making people immune means they cannot be infected with the pathogen and so they therefore cannot pass it on to other people. This is known as herd immunity. Examples of painkillers include ibuprofen, paracetamol, morphine, and codeine. Painkillers are taken to treat symptoms of diseases such as pain, inflammation, and to lower temperatures caused by fever. Painkillers do not treat the cause of the disease because they do not affect pathogens. Examples of antibiotics include penicillin, amoxicillin, and tetracycline. Antibiotics are drugs that kill bacteria. They treat the cause of the disease by killing the pathogen. Antibiotics cannot and do not work on viruses because these replicate inside cells and bacteria live outside of cells, so antibiotics cannot get into or work inside cells. Antibiotic resistance. Some bacteria in a population have the ability to survive treatment with an antibiotic. These will survive and reproduce, passing on the genes that allow them to survive to their offspring. Over time, we get an increase in frequency of the resistant bacteria in a population until we have a whole population of bacteria that cannot be killed by that antibiotic. This is an example of natural selection and therefore evolution of bacteria. We need to be able to suggest ways to reduce antibiotic resistance. We can reduce prescriptions of antibiotics for viral infections, so prevent doctors from giving out antibiotics unnecessarily. Making sure that patients take the full course of antibiotics every time, so they take all of the antibiotics they're given, even if they start to feel better. And reduce the use of antibiotics in farm animals. Originally, drugs were extracted from plants and microorganisms. There are 3 examples we have to know. One is aspirin, which is used as a painkiller, and this was extracted from willow tree bark. Digitalis, this is a heart medication that's extracted from the foxglove plant. And penicillin, which is an antibiotic, which comes from the penicillium mold, which was discovered by Alexander Fleming. New synthetic drugs made in the pharmaceutical industry can still have plant extracts as their base, but they must be tested for 3 things: toxicity, efficacy, and dosage. Toxicity is referring to whether or not it has serious side effects or is toxic to cells. Efficacy is whether or not the drug works to act in the way that we want it to, and dosage is about how much drug needs to be taken for it to work whilst not having too many side effects. We need to know the 3 stages of drug trials, what happens in each, and what they are testing for. The first stage is carried out in the lab, and it is known as preclinical testing, where we test the drugs on cells, tissues, and animals. At this stage, drugs are being tested for toxicity and efficacy. In the second stage, we move on to clinical trials, which involve humans. In clinical phase 1, we use healthy volunteers, and low doses are used to check for side effects. In clinical trial phase 2, we would use patients who are sick with whatever the drug is supposed to help with, and then we are testing for efficacy and dosage. Drugs cannot proceed through these drug trial stages unless they pass the checkpoints. So only if a drug is not toxic and found to work in the preclinical stages will it pass on to the clinical trials with humans, and only if the drug passes the clinical phase 1, where it's shown to not have serious side effects in humans, would it be given to patients, because it is unfair to give or risk patients' health or making them worse by giving them a drug that could cause serious side effects or that does not work. Clinical trials should be at least blind clinical trials. This is where we use a placebo or a fake drug as a comparison to show that the real drug is actually having the effect. In blind clinical trials, only the patients do not know if they are getting the real drug or the placebo. In double-blind clinical trials, neither the patients or the doctors know who is getting the placebo. This is the best standard of clinical trial. Double-blind clinical trials are important because they reduce the potential bias from the doctors. It reduces the psychological effects, for example, the placebo effect, where patients may just report feeling better because they think they are getting the real drug. And it can show that the real drug is having an effect when compared to the placebo. So we know for sure it is the drug that is working and actually having a measurable effect on the illness. Plants carry out photosynthesis using light energy absorbed from chloroplasts in the plant cells, carbon dioxide that they take in from the atmosphere as it diffuses through the stomata into leaves, water that is absorbed by osmosis from the soil by root hair cells, and that goes through the process of photosynthesis to produce oxygen, which is released into the atmosphere as it diffuses out of the leaves via the stomata, and glucose, which is then transported by the phloem to all cells to be used by the plant. Photosynthesis is the chemical reaction that takes place in plant cells that uses light energy, carbon dioxide and water to make sugars. The symbol equation is carbon dioxide plus water, light energy over the arrow, oxygen plus glucose, which symbol is C6H12O6. It's an endothermic reaction because it takes in light energy. We need to know how plants use the glucose they produce from photosynthesis. A lot of it is used up in respiration to release energy for processes in the cells. Some can either be converted to starch or used to produce lipids for storage. Some is used to produce cellulose in the cell walls. Some is combined with nitrates from the soil to produce amino acids. These can then be used to make proteins through protein synthesis. We need to know the factors that can affect the rate of photosynthesis. As you increase light intensity, it increases the rate of photosynthesis. As you increase the carbon dioxide concentration, it increases the rate of photosynthesis. Photosynthesis is an enzyme-controlled reaction, so increasing the temperature increases the rate of photosynthesis up until the optimum, and then it decreases if it increases past that optimum temperature. The amount of chlorophyll in a leaf or in the chloroplasts in plant cells, if there's more chlorophyll, then more light energy can be absorbed, so the rate of photosynthesis can be increased. We need to be able to explain what's going on in rate of photosynthesis graphs with the different factors. So for light intensity and carbon dioxide, the graph looks the same. There's an increase and then a flat line. On the increase or the slope of the graph at A, whatever is on the x-axis, light intensity or carbon dioxide, is the limiting factor, because as it increases, the rate increases. When we get to the flat line at B, another factor is limiting the rate of photosynthesis, not what's on the x-axis. So for example, it could be temperature. The rate of photosynthesis reaction with temperature on the x-axis looks very similar and can be explained in the same way as any enzyme rate of reaction graph. Initially, there is a low increase in the rate of photosynthesis when temperatures are low because there is less kinetic energy of the particles. Then we get to the peak, which is the optimum temperature and the fastest rate of photosynthesis. And then the slope of the graph goes down because higher than the optimum temperature, the rate decreases due to enzymes that control photosynthesis denaturing. These factors could be changed in a lab when doing the photosynthesis practical. or you could need to interpret how these factors might affect plants growing outside. When measuring the rate of photosynthesis in the lab, we can change various independent variables based around the factors that can affect the rate of photosynthesis. We can change the light intensity by moving a light or a lamp closer or further away from our plant. We should use a heat shield in order to make sure that only light intensity is being measured and not temperature. Different colours of light can affect the rate of photosynthesis, as more red and more blue light are absorbed and green light is reflected by chloroplasts. So we can use different colour LED bulbs or coloured filters in front of a white bulb. We could use a water bath at different temperatures to create different temperature conditions for our plant. Or we could change the carbon dioxide concentration by using different masses of bicarbonate of soda or sodium hydrogen carbonate, which we would dissolve in the water the plant is in. This test tube, you see, come up this test tube and measure that in a certain amount of time to be able to calculate rate. You could also use a gas syringe or a measuring cylinder to collect and measure the volume of oxygen produced in a set amount of time. Let's look at an example method with light intensity being the independent variable. The control variables will need to be anything other than light intensity that could affect the rate of photosynthesis. So we need to keep the temperature the same, the volume of water the pondweed is in the same, the carbon dioxide concentration, so the mass of sodium hydrogen carbonate used, the same, and the colour of light should be the same. The type of pondweed used or the same pondweed should also be kept the same. I've placed a heat shield, which is just a beaker of water in between the lamp and the pondweed, so that the heat energy from the lamp is absorbed into that water and increases the temperature of that water, but the light energy passes through to our plant. This means that only light intensity is the factor that's changing, and the temperature should not change with our pondweed. You could also use an LED bulb, which does not produce as much heat. My independent variable then is my distance from the lamp. As we move the bulb closer to our pondweed, the light intensity will increase. So we start far away, we measure the amount of bubbles produced in a set amount of time, then we move the bulb closer, we can use a ruler to measure the distance, leave the pondweed to settle for a small amount of time, at least 5 or 10 minutes, so that the rate of photosynthesis has time to change, if it's going to change. Then you measure the number of bubbles in a minute or however long time you're going to do again. And then again, you repeat by moving the lamp closer again, letting it settle, and then measuring the number of bubbles in a set time again. Just be clear in the exam what you are measuring, how you're going to measure it, especially having to remember to measure it in a set amount of time if we're going to calculate rate. Make sure you're clear about what the independent variable is, what are you changing in the experiment, and making sure that you understand that all the other control variables should be kept the same if they're going to affect the rate of photosynthesis. Respiration is not breathing. It's an exothermic chemical reaction that occurs inside every cell to release energy from sugars. Aerobic respiration happens in the mitochondria in every cell. It uses oxygen to break down glucose and release energy. The symbol equation is O2 plus C6H12O6, and then it produces carbon dioxide and water. We need to know the uses of the energy released from respiration in organisms. It is needed to carry out chemical reactions to build new molecules like proteins, carbohydrates, and lipids. It's needed for active transport to move substances against their concentration gradient. It's needed for movement, specifically muscle contraction, and also for keeping warm or maintaining certain body temperature. A reminder that any cell that carries out a lot of these processes, so active transport, muscle contraction in muscle cells, or building new molecules, they will have lots of mitochondria as an adaptation to make sure they can carry out more respiration to release more energy for these processes. Anaerobic respiration in animal cells takes place when there's little or no oxygen. It still breaks down glucose to release energy, but a smaller amount of energy is released. It happens in the cytoplasm of cells. So I've put in a box all of the facts we need to know about aerobic so we can start to compare it to anaerobic. So it happens in a different place. Anaerobic does not require oxygen. The products are different and the amount of energy released is different. In anaerobic respiration, glucose does not break down completely, but breaks down into lactic acid. Lactic acid is toxic if it builds up in cells, so it's removed by the blood to the liver. Anaerobic respiration usually occurs in muscle cells during high-intensity exercise, when there isn't enough oxygen getting to them. Anaerobic respiration in plant and yeast cells is slightly different, although in both cells it still occurs in the cytoplasm. Don't forget that in plant cells they also have mitochondria, so they can also carry out aerobic respiration as well. It still breaks down glucose to release a small amount of energy, but the products are different to anaerobic respiration in animal cells, and we call this process fermentation. Glucose breaks down to form carbon dioxide and ethanol. We need to learn about yeast and its anaerobic respiration because it's used in the food industry. The product of carbon dioxide from the fermentation reaction is used to make bread rise, and the product of the ethanol or the alcohol is used in the making of alcoholic drinks such as beer and wine. We need to be able to explain the body's response to exercise. During exercise, the body needs more energy from respiration. This is to help muscles contract. During exercise, the breathing rate and the volume of breathing increase in order to increase the concentration of oxygen in the blood. The heart rate also increases in order to pump more blood faster to muscle cells to deliver more oxygen and glucose. When talking about this, we need to use the word more a lot because your body always breathes and your heart always beats and you are always transporting oxygen and glucose around the body and your body does always need some energy. But when we are doing exercise, we need more energy because we're doing more muscle contraction, so we need to increase our breathing rate to have more oxygen and we need to increase our heart rate to transport more blood faster to the cells so they can have more oxygen and glucose in order to carry out more respiration. In muscle cells as well, we have a store of glycogen, which is broken down to release the glucose for respiration. If you have prolonged, high-intensity exercise, not enough oxygen can reach muscle cells eventually, and anaerobic respiration occurs in them and starts to produce lactic acid. The glycogen is also used up in muscle cells and they become fatigued and stop contracting properly. The lactic acid produced by anaerobic respiration in the muscle cells travels in the blood to the liver. The buildup of lactic acid causes an oxygen debt. The oxygen debt is the amount of oxygen needed to react with the lactic acid to remove it from cells. In the liver, the lactic acid is converted back to glucose. Needing more oxygen after intense exercise to clear the oxygen debt is why we breathe heavily after exercise, even when we've stopped. Metabolism is the sum of all the chemical reactions in the body. These are all controlled by enzymes and they require energy from respiration. Respiration itself is also a metabolic reaction. Photosynthesis is a metabolic reaction and it produces glucose. Glucose molecules can then be built together to make either glycogen, starch, or cellulose. Glucose can also then be combined with nitrate ions that plants get from the soil to build amino acids. Amino acids can be joined together to form proteins in protein synthesis. excess proteins in the body can be converted to urea for excretion in the liver. Glycerol and fatty acids can also be added together to build lipids. All of these are examples of metabolic reactions because they synthesize new molecules or break molecules down. I've highlighted in green photosynthesis, the conversion of glucose to cellulose, starch, or the combining of the glucose with the nitrate ions to make amino acids. All of these processes only happen in plants.
Ouch! This is why in some videos I explain scratches.